Hello and welcome back to part 3 of our look at the muscular system and muscle physiology. We had just spent some time just kind of refreshing your memories there, looking at the anatomy and the arrangement of thick myofilaments as well as thin myofilaments, the proteins that are involved with them, and how those two interplay with one another, and what is the role of ATP and calcium. And what we need to look at now is, beyond that, how does thin myofilaments and thick myofilaments engage with one another? And that is what you are seeing up here in skeletal muscle. And that is important to note here, that we are dealing with skeletal muscle. In skeletal muscle, we see this very organized manner where you have a thick myofilament, right? And surrounding that thick myofilament, you have one thin myofilament, 2, 3, 4, 5, and 6. And understand that 5 and 6, this is the adjacent, what we would define as being sarcomere. All right. Same thing with number 6. This is the adjacent sarcomere. All right. Now, what is a sarcomere? Well, a sarcomere is one complete unit of muscle arrangement. It's one complete unit of muscle arrangement. In other words, one complete set of thick myofilaments and the thin myofilaments that surround it. And so this right here is a sarcomere. Now, remember, one of the characteristics of skeletal muscle is that they are striated. And the striations come from the overlapping of thin myofilaments with thick myofilaments. And so there is an overlapping that happens between the two. And in fact, what we will learn is the overlapping of our thin and thick myofilaments is what is going to lead to contraction. That's what's going to lead to contraction. So let's go ahead and take a look at this, because there's some other things that we need to consider with the anatomy of our sarcomere. So let's consider the anatomy of a sarcomere. We know that it is a combination or an interaction between, again, the thin and the thick myofilaments, right? But how they are arranged is a little bit different. Right? And so the areas or the regions of a sarcomere that you need to be familiar with, first of all, are the A bands. And the A bands are the area where the thin and the thick myofilament overlap. And so this here is an A band. Right? Again, thin and thick myofilaments overlap. All right, so this is an A band right here, because we have both thin and thick myofilaments that are overlapping. Now, Where there are only thin myofilaments, where there is only thin myofilaments, that is what we define as being the I bands. And so where are there only thin myofilaments? Well, where we have only thin myofilaments are right here. Right. And so these areas right there, as well as right here, right, these are what we define as being the I bands. In other words, thin myofilament only. Right? I bands are the areas where there is only thin myofilaments. Where there is only thick myofilaments. Where there is only thick myofilaments. So in other words, this area right here, and that area right there, right, where there is only thick myofilaments, these are called the H zones. And you see that right down there. Right, and so the H zone is thick only. Thick myofilaments only for the H zone. Right. The M line, think of that as being an anchoring point for your thick myofilaments. The M line is what the thick myofilaments attach to. Right. That's what the thick myofilaments attach to. The Z disc is the connection between two adjacent sarcomeres. Right? It's the connection between two adjacent sarcomeres. So we have one sarcomere here, we have another sarcomere here, and where they connect, that is what we call the Z disc. Right? That is what we call the Z disc. And crossing over the Z disc, there is a protein called titin. Right? And so you see that right here, titin. Right? Titin are these little curlicue elastic bands that stretch across the Z disc. And what they do is they allow for, so as, let's say, this sarcomere contracts. Right? As this contracts, the titin expands like a spring. And then when the contraction is over, those springs come back together again. And so it allows for motion within between the sarcomeres so that you don't tear the muscle fibers. Because otherwise, if there was no give and one of those sarcomeres contracted, you could easily tear the adjacent or damage the adjacent sarcomeres. And so that's exactly what you see here on this slide. This slide here is just kind of reviewing, again, what those terms were that we pointed out on the previous slide. Now, what I would like to do is I'd like to spend a little bit of time going through the sliding filament theory. And the sliding filament theory is a 5-step process that in part reviews things that we've already looked at with the middle of our story. And so we're actually progressing now into the end of the story. We know how calcium is released. We know that this calcium goes into the sarcoplasm and it is allowed to attach to the troponin, and that kind of sets off this whole spree of events. But what else is happening? Because it's not just enough that calcium binds to troponin and ATP binds to myosin. What are the other details that are going through this process? And so that is what the next few slides are going to look like. And I will say that this is a 5.5-step process. And so what we are about to embark in is a 5.5-step process, and you might be asking, 5.5-step process, and you might be asking, well, how in the world did you get a half a step in? And the answer to that, is very simple. All right. And so if we look here, before step 1, all right, before step 1, aka before step 1, aka step 0.5. Right, here comes our half a step. Right. So what does everything look like? Well, what everything looks like here is, all right, ADP is bound to myosin during step 0.5. Right? ADP, adenosine diphosphate, is bound to myosin. Now remember, in order for muscle to contract, what must be bound to myosin? ATP must be bound to myosin. So right now, contraction is inhibited. It's blocked. It's blocked because we have ADP that is bound to myosin. So that's step 0.5. The other thing is there is no calcium present. No calcium present in the sarcoplasm. Why is that important? That is important because what that tells us is no action potential, is present, and calcium calcium ATPase has reabsorbed all available calcium from the sarcoplasm. All right, so because there's no calcium that is present within the sarcoplasm, no calcium is bound to troponin, which means tropomyosin is flipped, it's spun to block the myosin binding sites on actin. And the reason why there is no calcium present is because there is no action potential that has forced the opening of the voltage-gated calcium channels, and calcium ATPase has reabsorbed all of the available calcium from the sarcoplasm, making it impossible for anything to be bound to troponin. So that is step 0.5. Step 1. Neurotransmitters are released into the muscle cell. In other words, neurotransmitters are released into the sarcolemma. We will find out, I'm going to tell you now, but we're going to find out again, the neurotransmitter that we are dealing with here is what we call acetylcholine. Acetylcholine is the neurotransmitter that is going to initiate this process of releasing calcium. All right? So neurotransmitter is going to be released into the sarcolemma. All right? Part 2. All right? Calcium channel gates are going to open. And this is going to initiate an action potential. Right? Remember those junctional folds that we looked at in lab? So I'm going to kind of make a very bad sketch here. All right. So here's the sarcolemma. All right. Inside of the sarcolemma, remember there were these junctional folds. Right? And it was inside of those junctional folds that a neuron was innervating with. Right? So here is, it's the axon. This is the axon terminal. This is the sarcoplasm. Right, this is the sarcoplasm. Hold on one second, folks. Be right back. And so here is your axon. Here's the axon terminal. Here is your sarcoplasm. And right here are your junctional folds that are embedded into the, not sarcoplasm, sorry, into the sarcolemma, the cell membrane. And what will happen here is the neurotransmitter gets released into these junctional folds. And that is this is where your sodium channels are located. So your sodium channels, Na channels, are actually located in here. And that stimulates an action potential. So the acetylcholine binds to these sodium channels, causing the sodium channels to open, and an action potential is created, is generated. That action potential will travel from sodium channel to sodium channel to sodium channel to sodium channel. And every time it hits a new sodium channel, a new action potential is created. And this process continues down into the T-tubule. So this action potential is moving from sodium channel to sodium channel to sodium channel until it hits a voltage-gated calcium channel. And when it hits that voltage-gated calcium channel, calcium is going to be released. And that's exactly what we've looked at here in the beginning part of video 2. Right? We looked at this process. Right? We now understand that it's a neurotransmitter that is regulating that, acetylcholine. We understand that that acetylcholine is now binding to a sodium receptor in the junctional fold, and that is stimulating an action potential. We knew the action potential was there. We didn't know how it formed. We're starting to get a little bit better of an understanding of that. That action potential is traveling down the sarcolemma into the T-tubule, right, where it hits the voltage-gated calcium channel to cause it to open and to release that calcium. Calcium in the sarcoplasm is then going to bind to troponin. It's going to bind to troponin. Step 2. Step 2. Because calcium is bound to troponin, not troposin, that's a typo in the PowerPoint, to troponin, it's going to force tropomyosin to spin and uncover the myosin binding sites on actin. Right, so remember that actin is going to contain myosin binding sites. All right. And assuming ATP is now bound to myosin, actin and myosin will now bind. All right. And again, the warning here. All right. Assuming ATP is bound to myosin. So actin and myosin will bind, assuming ATP is bound to myosin and calcium has bound to troponin, which has now forced tropomyosin to spin, uncovering those myosin binding sites on actin. And again, ATP is what's stimulating myosin to reach out and try to bind to actin. Step 3, what we define as being the power stroke. All right. So what ends up happening now is myosin is going to flex that myosin sidearm inward. It's going to go towards, it's going to flex that myosin sidearm towards the M line. Right? So that myosin heads are now all going to flex towards the M line. Right? And when it does that, remember it's attached to the actin. So it's going to pull the thin myofilaments inward. And when it does that, it shortens the sarcomere. Now, I've got some videos posted for you that reviews that, so that way if you're visually trying to see that, you can kind of go back and watch the videos on that. This happens when ATP is spent. In other words, when we take ATP and convert it into ADP, that energy that is released powers the myosin sidearm to flex inward towards the M line to pull the thin myofilaments. That is what we define as the power stroke. And so we've used that ATP molecule. In step 4, that ADP molecule is released and a new ATP molecule binds to myosin. So the ADP that we just spent is released. In step 4, a new ATP molecule binds to myosin, and that leads to myosin unattaching from actin. It releases actin. In step 5, ATP is now broken down. All right, which means when we say cleaves, it means it breaks it down, and if we're breaking ATP down, we're creating a DP plus that inorganic phosphate. And so we've created now another ATP molecule, or ADP molecule, and what that does is it forces the myosin sidearm to swing back into its original place. And so we've forced the myosin sidearm, we've reset the myosin sidearm back to its original starting point. And so let's kind of play this out. What is attached to the ADP molecule, what is attached to myosin at the end of this process is ADP. At the end of step 5, ADP is attached to myosin, and the myosin sidearm is back in its original place. Right? So what happens next? Well, if calcium is still present in the sarcoplasm, that means that calcium is still bound to troponin, which means tropomyosin is still in a position of uncovering the myosin binding sites on actin, and so another ATP molecule could bind to myosin. And step 1 happens all over again. And step 2, and step 3, and step 4, and step 5. If calcium has been returned to the terminal cisterns via the calcium ATPase pumps, then there is no calcium bound to troponin, which means tropomyosin has spun back, covering the myosin binding sites on actin. And so tropomyosin is now inhibiting actin and myosin binding. ATP is not bound to myosin, so that is inhibiting myosin from binding to actin. And we simply wait. And we wait for a new action potential, because a new action potential triggered by acetylcholine being released in the junctional folds is going to open the sodium channels in the junctional folds to initiate an action potential. The action potential is going to travel down the sarcolemma, down into the T-tubule, where it's going to go ahead and it's going to run across a voltage-gated calcium channel that is going to then cause that calcium channel to open, releasing calcium from the terminal cisterns into the sarcoplasm, where that calcium will bind to the troponin, which is then going to prompt tropomyosin to spin, uncovering the myosin binding sites on actin. ATP will bind to myosin. ATP will be spent, which will then allow actin and myosin to bind, that ADP is released, a new ATP is attached after the power stroke, which will release myosin from actin, creating ADP, which is where we are in step 5. Right? That is the sliding filament theory. All right? The sliding filament theory, so named because of how that thin myofilament slides over the thick myofilaments, directed by calcium and ATP. And that is how your muscles contract. Now, of course, there's a little bit more detail to this that we will look at in video 4, specifically what happens with the action potential. But these are the steps that are involved with muscle contraction. It's a lot for you to chew on, so I'm going to allow you to chew and review and answer some questions. I'm sure you will have questions for me. And so with that, I say, I'll see you on the flip side.